Hello.
Hello.
Our next speaker will be Dr. Agata Kołodziejczyk. She will tell us about the use of bacterial cellulose in regolithic substrates of plant cultivation. It will be about testing new substrates for the food production in space. So the floor is yours.
Thank you. Good evening, everyone. Good afternoon, depending on your time zone, and maybe, yeah, good night. It's a pleasure to be here. Thank you very much for all this organization of such a wonderful conference. And today I'm going to speak about kombucha. I'm really happy that I'm after Monica because our topics are kind of common. I will speak about application of kombucha not only for using as a medium for growing food, but also for some decorations, as you can see here on my earrings. And I will also show you something what you can build out of kombucha. So let's try to share my screen. Let's see. I think that here should be fine. And now let's make the slideshow. I hope that you see it properly. Yes?
Yes, we can see it.
Great. So yes, my presentation will be mostly about several types of biomaterials that I was producing within several teams. You can see here that there are several universities and companies involved, and still we are doing many interesting studies together, mostly focused on applications, so on development of technologies and thinking that we want to apply them, including space. So, where am I? As you know, kombucha is something that you can buy in the shop, and it is something for drink. Usually in space, you are not allowed to drink kombucha because it contains some bubbles and it will cause pain in your stomach. You cannot burp these bubbles, you need to vomit them, so it is not nice for drinking. But here we will focus on something that is a bioproduct of this consortium of bacteria and yeasts, which is bacterial cellulose. And you can see it is growing on top of the solution. When we compare the properties of bacterial and plant cellulose, we can see that bacterial cellulose is really great biomaterial. What is another very interesting thing is that it is very abundant. Bacterial cellulose is produced by several species, you will see in a moment. So we can see that, for example, degree of polymerization is quite high. The purity is fantastic. The water holding capacity is also something what later on we will use. I will show you how we can use these parameters to make interesting materials out of it. Here I showed you that there are several bacteria. They are mostly acidic bacteria that are producing bacterial cellulose. And you can see that depending on species, you can get a different structure of these cellulose. Either these are fibril structure or ribbon structure or amorphous or even not defined. In one of our last papers, written down by several scientists over the world, including people who are running experiment in space on International Space Station with kombucha, we wrote a paper that there are several dimensions of applications of this material. So either it can be used for food and packaging. By food, I mean that it can be even used as something like a fibers to clean our guts. So this cellulose, of course, will not be digested by us, by humans, but it can be also a part of some bioproduct that we can produce as a bioreactor. Then, of course, this bacterial cellulose can be used as bandages, as some prebiotics, something that has a medical application, and of course some products like cloths, lenses, like fuel cells. So this is very important because we need to develop such artificial consortia in space that can, first of all, be sustainable, that can resist all different types of harsh conditions, that they can be productive, that they will do what we want. And in several years, I was working on kombucha, starting from European Space Agency, where I was a postdoc in biomimetics. And I was trying, for example, to create ropes, because you know that until now, there is no technology, in situ technology for space application, how to create ropes in space, especially on the moon or somewhere on the spaceship. We still don't know how to create ropes, and ropes are very important tools, very important parts, elements of our life, of base, the future base. Also, we were thinking about how to create 3D shapes. And it means that, for example, each bacterial cellulose is different because it grows in different conditions. It is thicker and thinner. It is not same structure everywhere. So how to homogenize the structure? Of course, we need to create something like OSB plate from wood, from plant cellulose. So we need to unify the material. And of course, we have been thinking about this unification, how to make this material able to shape forms that we want and not that bacteria are doing. Of course, this is one thing, 3D shaping, and of course, possibility of 3D printing. I will tell you in a moment what are our actual studies. Then we are trying to build composite materials. So how to combine, for example, bacterial cellulose with carbon nanofibers or mexins, which are very interesting materials that can sense. They can be used for smart materials. Yes, they can be sensors. Of course, kombucha can be used for packaging, and already now, even at your home, you can use it to cover some fruits and put them to the fridge. This is a great protector because it contains antibiotics. It is really preventing from some biocontamination. So you may try it at home, and if you want to have it clean, it is very easy just to boil it for 15 minutes, and you have a pure packaging material ready to reuse again. So you don't need to use plastics, you don't need to use foils and to contaminate your body by microplastic, yes? Of course, from kombucha we tried to do paper. Well, we live in the area, you know, in the time of laptops on digital matrix, so we don't need paper so much. But if somebody wants paper, still can be produced from this wonderful biopolymer. And here you can see some beautiful images from electron microscopy we did with my collaborators at Jagiellonian University, and you can see these small bodies that are producing the cellulose. On the left side, you can see the raw material. I'm speaking about this photographies in the upper row. When you lyophilize this hydrogel, because this is hydrogel, you can see removed water and only fibers. This material is very weak, it is very crunchy. But if you coat it, for example, with glycerin, it can be very simple coating. It will keep elasticity, it will be very nice for use. And you can see also the coated surface. We tested if this material can be used, for example, in space for astronauts as cloths. Of course, then you need to have anti-flammable materials, because in space, fire is something that you don't want, and especially cloths, they must protect you against this. So we observed that our coated cellulose is very good protection from fire. Also, we were trying to analyze what kind of situation can be observed when we soak this cellulose in fresh water and in salt water, say in the sea or in the ocean. And we could see that when you take it out, until it is soaked, it still has very nice properties that it can be used underwater. But then if it gets dry, you can already see on the left side, lower row, you can see some crystals of salts that are inside this matrix. And here is our hero, the acidic bacterium producing beautiful polymers of bacterial cellulose. Here you see that this consortium is full of other organisms that live together, and this symbiotic relation allows kombucha to sustain so resistant and so kind of stable. Why I'm saying kind of? Because they observed on ISS, it was an experiment for 18 months, 8 months, where this kombucha was exposed on the Kibo panel directly to the space conditions, and they observed that some species of this consortium that they die. But fortunately, bacteria that are producing bacterial cellulose, they are not dying. They are super resistant. So here you can see more beautiful images and even higher magnification. First time we tested bacterial cellulose as a cloth, as a possible, part of astronaut clothing in one of our analog missions. It was a Spectra mission in Poland, in Lunaris habitat. You can see Cyan Proctor, and until now I still have her brand slide that was tested during this mission. So we produced this elements from kombucha cellulose, and we asked our crew to have it on their wrists for 2 weeks. And we were trying to see if there will be some allergy, all types of possible reactions with the body. And what we could see, you have the fresh wristbands on the left, and in the middle you see the wristbands removed just after the mission, and then they can be recovered to the initial shape. They were, soaked in water. Crew was wearing them all the time. And also we tried this t-shirt. You can see some incrustations, some elements of kombucha cellulose, and these parts were designed where people are sweating mostly. So the areas that people sweat mostly, and we tried to see how kombucha will react with these parts of the body. And of course,
just 2 minutes left, if I may.
Thank you. Yes, great. So why we try to wear kombucha? It is because we want to insert kombucha in a broad cycle in isolated space concept. So we want to have kombucha everywhere in each level of ecological chain inside the base or inside the closed system that will must be somewhere on the moon or on Mars or even farther. Yes, so it must be somehow sustainable. We observed that, of course, in space there will be hard access to have a tea. So we tried to cultivate bacterial cellulose using only algae. And we know that chlorella already, they are used in space, and they are implemented in several closed systems that are now tested in the world. So now we can produce bacterial cellulose without tea only with chlorella. And now our challenge is to reduce sugar, sucrose, which is a polysugar. We want to exchange this polysugar for some basic sugars, or even different chemicals. We are trying. Fortunately, kombucha is an artificial consortium. This is not occurring in nature. Yes, it was done some very, very long time ago by humans, we guess. So now we are modifying this symbiotic consortium in order to get what we want. And what we want? We want, of course, to have several types of biomaterials. So what we test? We test different types of methods of production cloths. You can see on the left, one of our students is using several kind of chemicals to make this material colorful in this case, or to have stronger parameters, or to be more elastic, many things. Here on the right side, you have very funny way of application of kombucha. It is to use them as masks, hydrogel masks, to reduce pimples and all noise on the skin, let's say like this. Of course, kombucha is also well known to be used now as a bioactive cosmetic. And even I brought you here for this presentation a cream that I'm producing. These all products are produced by our people here, and this contains a live kombucha, which is completely different what you can find in shops. And well, I think that each person, including astronauts, can create something like this, and it is really good. Finally, with AGH here in Krakow, we are collaborating with people responsible for 3D printing. And actually, we are on very interesting state because we can already use kombucha as a glue for regolith. So we understand that first phases of colonization of moon or other celestial bodies will be a robotic stage where there will be a big machines 3D printing the basic skeleton of structure. And after this phase, humans will come and they will try to develop the second and other stages of settlement. So we now are trying to assume that this first stage will be robotic, and we try to optimize 3D printing using lunar regolith in this case, and using kombucha as a glue. I told you already, we try to grow our bacterial cellulose with different types of nanomaterials in order to produce sensors, biosensors. This is very interesting experiment that we did a couple of weeks ago, where we glued kombucha with lunar regolith simulant. You can see different diameters of this lunar regolith simulant. And we observed the growth of cress on these different types of medium. So we can see differences and very interesting results from very thick granules, what you can see in the middle lower row. Yes, and we already can see that this cress growth, the biomass production, was the highest at this the large diameter granules, comparing even to control to the soil. So it is very interesting result. Of course, with kombucha, we can try different types of shocks. We are torturing this material, unfortunately, but we want to understand its properties. And of course, as I told you already, we are still working on modification of this consortium. One of extreme environments that we test is the stratosphere. We are first generating these kombucha sensors, the composite materials, and we are sending them to the stratosphere to feel to sense several parameters. With that, I wanted to thank you, but I have some how to say, surprise for you. I don't know if still I have time. This is like a 2 minutes movie.
There are only 2 minutes left for discussion. And
2 minutes left for discussion. Yes. Only.
Yeah. Well.
Okay.
We have one question on the chat, so well, you can show us your surprises.
I can show you your surprise.
Yeah.
My surprise.
You still have some time.
You.
Yep.
Thank you so much. So I'm showing.
But briefly.
Yes. Yes. So I wanted to show you a movie from the last mission, how our analog astronauts, how they were preparing this experiment with kombucha and regolith. This is a universal mission. And of course, it was in the habitat in a simulated lunar base.
Are you sharing screen or something? Because we
You don't see it?
No, we don't. We can't see it.
Oh.
The screen is not sharing.
I think I know why. But I will do like this. I know why. Because I have already the other window, but don't worry. I will not go. Oh, it is the proper moment. You see here. Maybe I will start from here. Now you can see it.
Yes, we can see it.
Exactly. This is the part of future agriculture with kombucha, with regolith. And everything inserted, of course, in the isolated, because on the moon we have to create everything basically. So maybe with this I will finish because I want to answer the questions if you have.
Yes, there is a question from Camille. Does it mean that Freeman Dyson was right and human space exploration will include greening the galaxy?
Can you please repeat, because I didn't get the first part of the question.
Okay, does it mean that Freeman and Dyson was right and human space exploration will include greening the galaxy?
Wow, this is some citation of some artist. Well, definitely, I believe that we humans are able to terraform other lands, and this is the challenge of astrobiology, I believe, of synthetic biology, what Monica previously said. This is the challenge for us, our generation, and future generations to make this working, and I believe because of that, of this turning the eye to the microbial world, which is still terra incognita for us, I believe that this will dramatically change our perception of the world, our perception of galaxy.
I see. Thank you for the answer. Maybe I will have another question. I don't see any other questions, so probably it will be the last one. So does the microgravity or gravity on Mars, could it disrupt the production on kombucha? Could it have some effect on it? Because, well, our gravity on Earth is different than on Mars on the orbit. So
absolutely, microgravity will have effect on several enzymes. This we know already from microgravity studies. But there is one very interesting but. Scale of these organisms is small enough in order to make some simulators of gravity. So it will be easy to create like centrifuges that can be used to generate gravities sufficient to provide proper growth.
I see. So we just need centrifuges on Mars.
Anyway, they have on ISS because each experiment should have control. Even on some orbital experiments on satellites, they always have some artificial gravity induced for our organism in order to have reference.
I see. Thank you. And well, if there are no other questions, I would like to thank you one more time for a presentation and for video.